Hi, my name is Beth Pitel. I'm a senior developer in the Mayo Clinic Genomics Laboratory, and I'm excited to share with you today some work we've done on creating a standard procedure for curating and maintaining cancer-specific gene lists. And this is done as a collaboration between the Cancer Genomics Consortium and the Mayo Clinic Genomics of Oncology annotation team. I have nothing to disclose. In this presentation, we'll cover a couple different topics. So what exactly is a cancer-specific gene list? How do we make it, and what is it useful for? We'll describe the collaboration between CGC and the Mayo Clinic GOAT, and also define the essential elements of a gene list. So what exactly needs to be in a gene list in order to make it useful? Lastly, we'll talk about clinical significance levels, which are a similar concept as AMP-ASCO-CAP tiers are to variants, only this is on a gene level. A cancer-specific gene list is essentially a list of genes that are important within the context of a specific type of cancer. And in this gene list, we provide important gene information. So what's the mechanism of alteration? What are common fusion partners, pathway information, diagnostic, prognostic, therapeutic information associated with variants in each gene? And the purpose is to gauge the maximum clinical relevance of a gene. So we know which genes to focus on when we're looking at high throughput assays, like chromosomal microarray, whole genome sequencing, whole exome sequencing, large panels, and we'll also provide relevant references. So a little bit of information about the Mayo Clinic GOAT. We are the Genomics of Oncology annotation team, and really this team arose out of the necessity when we went live with neoplastic chromosomal microarrays. As many of you know, these tests are often exceedingly complex. So what this group did was we looked at what kind of cases were represented in the lab. We did deep literature dives into knowledge bases and WHO books, and what arose were 26 disease-specific lists that are represented in teal here, as well as a pan-cancer list with over 2,200 genes. In previous CGC meetings, we have shared our ALL gene lists, and there has been interest in sharing more of our lists. And we decided to do this through a standardized mechanism of expert review through the CGC working groups. So the GOAT and CGC working groups is really an ideal collaboration. CGC disease-specific working groups provide expertise that the GOAT team does not necessarily have, and as well as a multi-institutional representation that offers a lot of value in making these lists robust and applicable across many different institutions and many different viewpoints. So the CGC currently has 9 working groups. You can see that in the middle panel here. And there is room for more working groups. So if your disease of interest is not represented here and you are interested in starting up one, you can recruit your colleagues and friends and get in touch with the CGC Board of Directors. So before the CGC cancer-specific working groups actually started curating these gene lists, we decided to look at what are the essential elements that need to be included in every gene list. And we decided on just a few, and those are listed on this slide, and include gene name, the type of genomic alterations observed, the functional mechanism of pathogenic variants within that gene, clinical significance level, which we'll talk about in a couple slides, and then also peer-reviewed references. So these are the core elements that are necessary annotation for all genes on the gene list. In addition to essential elements, we also defined optional elements, which can be represented in gene lists and might often be really helpful, but are not needed in every gene list. And those elements include additional information describing clinical relevance of each gene, genomic location, disease subtypes, and association with germline predisposition. So clinical significance levels are a newer concept that we came up with, defining what the maximum impact of a gene or a variant within a gene is in the context of a specific disease. And the purpose of having these tiers is to gauge the maximum impact that a variant in a gene might have. We'd like to catch every variant that is clinically significant in our assays. And so we came up with 3 clinical significance levels that include high significance, moderate significance, and emerging evidence. A high significance gene is a gene with specific or characteristically enriched within a tumor type per guideline. So these are genes that are included in WHO and NCCN guidelines, ESMO. Often these are disease-defining genes. These are genes that harbor germline pathogenic variants that are associated with clear cancer predisposition, and these are genes that are actionable or therapeutically relevant. Some examples might be BRCA1 in breast cancer or FLT3 in acute myeloid leukemia, where these are prevalent in guidelines. BRCA1 obviously has germline implications, and these also have therapeutic implications. Significance level 2, so genes with moderate significance include genes that harbor recurrent abnormalities. They're probably pathogenic and they have significant evidence of being associated with a cancer type, but they're not targetable. They're not diagnostic, prognostic, or part of guidelines as of yet. So one example would be CRE-BBP in acute myeloid leukemia. We know the mechanism of the alterations, but we really don't have any clear knowledge of this being actionable, diagnostic, or prognostic. And then there are genes with emerging evidence. So these are genes that have not enough evidence to presume pathogenicity within this disease type. So one example is HMGA2 in myeloid malignancies. Often fusions in HMGA2 are activating. However, we don't know enough about this gene in the context of myeloid malignancies to call it pathogenic. So now that we've defined the elements of what needs to go into a gene list, we can start curating these lists. And so what has happened is genes are assigned to experts that check the relevance of the gene, check the mechanism of variants within that gene, and evaluate the reference list. Each gene is reviewed by at least 2 independent reviewers, and there are meetings for the working groups to discuss any discrepancies between reviewers and to come to a consensus. Gene lists are currently in spreadsheet format and bed file format. Bed file format is useful for incorporation into analysis software and genome browsers. Additionally, the CGC working group members can identify genes that should be considered for addition to the gene list as well. So now that we've collaborated with the myeloid and the breast cancer working groups, we're realizing that our work is just getting started. We have plans to expand this collaboration to more cancer types and essentially more CGC working groups. And we'd like to also expand the accessibility of these lists and incorporate them into the CGC website and then potentially into the CCGA. Additionally, we have a pending manuscript that we plan to submit within the next couple months. There are so many people to thank for the success of this project. Thanks to the CGC to being open to this collaboration. Thank you to the Mayo Goat team, both members past and present, whose work has really helped shape the art of making a gene list. And then also thanks to the CGC Education Committee and to both working groups who worked really hard on making these gene lists really high quality. So really grateful for this excellent team. Thanks so much for listening to this presentation. Of course, if there are any questions, feel free to email me, pitel.beth@mayo.edu. You can also find me on Twitter or LinkedIn. Opinions are my own. So thanks and have a great meeting. Bye-bye.